Hello and welcome to another lecture video on cognitive psychology. For this video, we will be discussing about cognitive neuroscience. So we would have to see about different concepts of the brain and functioning. Our brains are a central processing unit for everything that we do. But how do our brains relate to our bodies and how are they connected or are they separate? And so for this lecture video, we will be discussing about the different structures and the different functions of our central nervous system. So the mind-body issue has long interested philosophers and scientists. So before, people thought that the mind and our body are actually separate entities, but later on, because of recent researches, we have um, learned that our mind and our body are actually intertwined with each other. And so we would understand about how the mind and the body interact. So this chapter outlines the different concepts on the anatomy of the brain, the neuronal structure and function of the brain. We would also get to review and discuss about the different neurotransmitters and its functions. We would also have to discuss about the different ways or methods of viewing the structure and functions of the brain. And lastly, we'd also tackle some brain disorders that disrupt cognitive function. So this chapter, which is cognitive neuroscience, would study how the brain and other aspects of the nervous system are linked to cognitive processing and ultimately our behavior. So to begin with, let us have a brief review about the anatomy and the mechanisms of the nervous system, or the central nervous system to be precise. Now, our nervous system is an important component in our brain because it is only through our nervous system that we're able to perceive, adapt, and interact and navigate within our surroundings. So it is because of our nervous system, or the communication of our nervous system, that enables us to plan, to solve problems, to be creative, in order for us to properly navigate our environment. The brain, as we all know, based on your learnings from your physiological psychology class, is the supreme organ of the nervous system. So this is where most of the complex processing are happening. No? So you would, if you recall your discussions on physiopsych, you would understand that most of the things or processes in the peripheral nervous system are just mainly communicating the sensory information, no? But it is actually in our brain where it is being processed. Okay. So we would look into different parts of the brain, and we would review or recall that different parts of the brain have its own functions and its own controls on cognitive processes. So now let's have a look at the structure of the brain. So our human brain is divided into three major regions. So you have your forebrain, you have your midbrain, and you also have your hindbrain. So first, let's talk about a bit about the forebrain, or this is the part of the brain or the major division of the brain that is responsible for complex and higher order cognitive processing. So it is involved with receiving and processing sensory information, thinking, perceiving, producing and understanding language, and controlling motor function. So it is because of our forebrain that enables us to what we are as human beings. No? So it is in these regions where complex cognitive processes are happening. So this is what makes human beings humans. Now let's go to the next major division or major part of the brain, which is the midbrain. So the midbrain is in the middle part of the 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 brain, and the midbrain is actually interconnected with the hindbrain. So the midbrain and hindbrain is actually working together um, to make up the brainstem. So the midbrain is responsible for perceptual processes such as auditory and visual responses, as well as motor function. So they also aid with the processing of these sensory information. And lastly, you have the hindbrain. So the hindbrain is the one connected to your spinal cord. And so this is the first part of the brain or region of the brain that receives the information from the other parts of the body, from sensory information from your hands, from your feet, and etc., etc. So this is where it receives the information coming from the spinal cord. So if you would recall your physiological classes or lessons, rather in physiopsych, you would recall that most of the information, except for those carried by cranial nerves, would have to first go through the spinal cord before it can be received by our brain. So the hindbrain, parts of the hindbrain are responsible for life-sustaining functions, for breathing, for arousal, etc., etc. So the regions, for example, your pons, your cerebellum, are responsible for maintaining balance, equilibrium, movement, coordination, and conduction of sensory information, and other, again, essential for sustaining life, such as breathing, your heart rate, digestion. So if if there will be damage to your hindbrain, so it would most likely automatically lead to death. Again, because the lower the part of the structure in the brain, the most basic its function in maintaining life. And the higher it is in the hierarchy or the structure of the brain, then the more complex the processing are happening in those regions. Okay. So here are the different parts of the forebrain. And later on, we'll also see the midbrain and the hindbrain. So part of your forebrain, or the structures included in your forebrain, are your cerebral cortex, and later on we will discuss more about the cerebral cortex and its different structures. You also have your basal ganglia, the limbic system involved for memory and emotional and motivational related processing. You also have your thalamus and your hypothalamus. So these structures have their own distinct and specific functions um, in your system. For example, for the cerebral cortex, it's responsible for receiving and processing sensory information. It's responsible for higher order processing. Basal ganglia is responsible for motor movement, for the limbic system, for learning, emotions, and motivation. The thalamus is responsible for, aside from it acts as the relay station in the brain. No? So, for example, um, in the visual perception or visual processing, before the sensory or the visual information is being processed in the primary visual cortex, it has first to pass through the thalamus, no, before it can be delivered to the visual processing cortex. You also have your hypothalamus, which is involved in the endocrine system, responsible for the secretion of hormones. It has also something to do with your autonomic nervous system, survival behaviors, and etc. etc. So these are the different parts of your forebrain. Your midbrain is composed of the superior calculi, inferior calculi, and the reticular activating system. So it also has its own functions in our system. For example, the superior colliculi is responsible for vision, while the inferior colliculi is responsible for hearing. Your reticular activating system is also important in different functions such as consciousness, attention, um, cardiorespiratory function, and movement. Then let's have your hindbrain, or the part of the brain that is directly connected to the spinal cord, as I said earlier. So parts of the hindbrain include your cerebellum, your pons, and your medulla oblongata. So for your cerebellum, it's essential for balance, coordination, and muscle tone. So this also, the shape or the structure of the cerebellum also mimics that of the cerebral cortex. So thus, it is the little brain in the hindbrain. Your pons are responsible for bridging other structures in the brain, and it is involved in um, consciousness, facial nerves, and again, bridging neural transmissions from one part of the brain to another. So again, the pons serves as a bridge, no? if you can still remember that. And you also have your medulla oblongata. So this part of the hindbrain, it is where nerves cross from one side to the body to opposite side of the brain. It's also responsible for cardiorespiratory function, digestion, and swallowing. So here are the different structures, the major divisions of the brain structures and its corresponding brain structures and functions. So this is would just serve as a review because this was already discussed in your previous classes. Okay. Now let's talk more about the cerebral cortex. So the cerebral cortex, it's very essential because it plays an important and essential role in human cognition. So if you could still remember the case of Phineas Gage, it was actually the cerebral cortex that was damaged, and therefore it led to a lot of cognitive deficits. No, so Phineas Gage, when he suffered a damage on the cerebral cortex, it also affected his planning, his emotion regulation, and etc. etc. So it's because of our cerebral cortex that makes us human beings. No? It enables us to um, do and enables us to participate in higher order and complex cognitive processing. So it includes planning, it includes coordinating thoughts, it includes our actions, um, perception of visual and sound patterns, the use of language, production of language, and these are very necessary cognitive functions in our everyday living. No? So it is necessary for your studies, for your work, for communicating with other people, and um, other activities and tasks that we may do in our everyday living. So without our cerebral cortex, so we would not be humans. Um, So as I've said earlier, the cerebral cortex is again responsible for these complex cognitive processing, and one phenomenon that we can also see or we can also observe in the cerebral cortex is what we call the hemispheric specialization. That means certain parts of the cerebral cortex are responsible for a specialized function. For example, the Wernicke's area and the Broca's area, which is responsible for speech or language. So it's involved in speech production and speech comprehension. So damages to these area would also mean that there will also be deficits on these functions in language or producing language or understanding language. The cerebral cortex is further or can be divided into different lobes, and each lobes tend to have their own function. They tend to have or specialize in um, different functioning. So you have the four lobes. You have the frontal lobe, you have the parietal lobe, you have the temporal lobe, and the occipital lobe. So again, these concepts will just serve as a matter of review because these were already discussed in physiological psychology class. So let us first familiarize or rather review the different lobes. So as you can see in the picture, you have the frontal, the parietal, the temporal, and the occipital lobe. Now let's recall the different functions of the four lobes. Okay, so again, earlier I said that the cerebral hemisphere is or can be divided into four parts, but they are not necessarily distinct units as if they work separately from each other. But again, they are just largely arbitrary anatomical regions divided by fissures. But again, they still interact or they still work together. No? So for example, even if your occipital lobe primarily processes the visual input, but again, the information being processed there would still be used for other functions. For example, when you are walking, there will be simultaneous cognitive processing. And so even if there are different areas responsible for processing, they are still integrated to become a more coherent information. So again, they are not really distinct separate units, but they're interdependent regions of your cerebral hemispheres. So they interact with each other. So the frontal lobe, as a recall, it's involved in reasoning, motor control, emotion, and language. So it contains motor cortex which is involved in planning and coordinating movement. So this is where these certain information are being processed in your frontal lobe. No, when you talk about motor movement, when you talk about language, reasoning, emotion, planning, etc., etc. So higher complex processing are processed or information are processed in the frontal lobe. Then you have your parietal lobe, which is responsible for processing of your somatosensory information coming from your skin, from your hair receptors, no, pain receptors. So this is where it's being processed. You also have, aside from, so again, somatosensation, sorry, refers to sensations such as touch, temperature, and pain. You also have your temporal lobe, which is associated with first your audition or hearing. So your primary auditory cortex is located in your temporal lobe, so near your ears. So aside from hearing, it is also responsible for memory, for emotion, and some aspects of language. So it is here where most of the auditory information are being processed. And then you also have your occipital lobe, which is at the back of your head. It contains the primary visual cortex. So most of the processing of visual input, no, from a certain stimulus or stimuli are processed at the back of your brain or your occipital lobe. So this is responsible for interpreting incoming visual information. But then again, if you can still remember your concepts of perception, again, so there are different hierarchy of cognitive processing. No? So you have your primary cortex, you have your secondary cortex, and then you also have your associated cortices. So there are different also, there are other different regions of the brain that are also part of, for example, visual processing that is not necessarily part of the occipital lobe because again they interact with each other. Okay. But before we could fully understand about these different regions of the brain or different parts of the brain, we also need to understand its basic or um, fundamental structure. No? So which has something to do with our neurons. So if you could see, there are two specialized cells, or if you could remember, there are two specialized cells in the brain. So one is your glial cells or glia, and then there are also different classes of glial cells. Now you have your astrocytes, you have your Schwann cells, you have your microglia, and these different types of glial cells, they also have their own different functions. No? You would if you can recall, your glial cells are responsible for myelination, responsible for the engulfment of debris in the central nervous system. It's responsible for supporting and providing structure to new neurons, and etc. etc. So that's one specialized cell you can find in your central nervous system. But you also have your neurons, which are responsible for the receiving and um, transmission of different signals, no, from one neuron to another. So before we could fully understand about the entire nervous system, we also need to examine the basic structure and functions of neurons and of course glial cells that comprise the nervous system. So if you could still recall again, each neurons or neural cells, they transmit, they receive electrical signals and they transmit chemical signals, which will later then be again triggers another electrical signal. So if you could recall the two modes of communication in the nervous system, you have your neural conduction, which is within a single neuron, and synaptic transmission, which is the communication between two neurons. So, synaptic transmission, although neural conduction is, essential also in the communication in the nervous system, but synaptic transmission is a complex process that occurs in our nervous system. So it is the communication from one neuron to another or neighboring neurons. So it is actually here during synaptic transmission when or where information are actually sent, until it's fully processed, from the sensory organ until the brain. And we also need to recall that there are different processes or different stages in neural conduction and synaptic transmission. For example, in neural conduction, action potentials happen during the neural conduction process because, again, it's the electrical signal that is being um, received by the dendrites, then it's being passed down towards the rest of the body of the neuron until it reaches the axon, and then the axon buttons or axon terminals, which will then trigger the next mode of communication, which is the synaptic transmission. But because neurons are not directly attached to each other, they just stay very, very close in junctures, no, or in synaptic clefts. So they're not really attached to each other, or they don't touch each other. So the electrical signal being conducted from one neuron must be able to first be translated into a chemical messenger, because, again, electrical signals could not jump from one neuron to another because they are not attached, and so the chemical messenger will be secreted instead, and when it binds to the neighboring neuron through its receptors, then it triggers another action potential, thus leading to another neural conduction. So, and that's how basically how neurons communicate, no, by neural conductions and synaptic transmission. So, um, looking at this image, no, so let's just try to recall the different structures and function of a neuron. So you have your dendrites, you have your soma, which contains the nucleus and the other parts of a cell. You also have the axon, and you have your axon terminals. So neurons could either be myelinated or unmyelinated. So we all know that myelinated cells or neurons, they tend to transmit signals faster compared to unmyelinated ones, because myelinated neurons, they use saltatory conduction in transmitting the signal from the dendrites until the end of the axon terminals. So again, once it reaches the axon terminals, it triggers the release of neurotransmitters, which are already being packaged, inside a neuron, so it could be delivered to the neighboring neuron. So those are the major modes of communication within the nervous system. So we'll highlight more on the synaptic transmission part, because, again, it is crucial also in the communication among neurons. So because neurons are not directly attached, they need chemical messengers, or we also call them neurotransmitters, that transmit information across one synaptic gap to the next neighboring neuron. So there are a lot of different neurons identified. Sorry, there are different neurotransmitters identified rather by scientists. But it seems like there's still more that remains to be discovered, no? But for now, we will stick with the common ones, neurotransmitters. And so neurotransmitters are, it's essential for us to understand about neurotransmitters because, again, they interact with drugs, they also influence our mood, our abilities, and our perception. So it's actually because of these chemical messengers that allows us to perform these functions. So we have here 8 neurotransmitters. We have adrenaline, noradrenaline, or you could also call this epinephrine for adrenaline and norepinephrine for noradrenaline. So it's the other name for these two neurotransmitters. And these two neurotransmitters are responsible for when an individual is involved in a vigorous activity. For example, adrenaline is responsible for the fight or flight response. For noradrenaline, it's also used when somebody is engaged in a physically demanding activity, for example, sports or doing manual labor. Dopamine is another neurotransmitter. which is responsible for pleasure. No, it has something to do with the reward system of the brain. It's also responsible for motor movement. It's also associated with attention, learning, and again, movement coordination. So, if an individual would have an excess amount of dopamine or a depleted amount of dopamine, it also results to deficits. No? So, for example, if the dopamine is too much, if it's too high, then the individual may manifest symptoms of schizophrenia, no? Responsible for um, characterized by delusions, hallucinations. But if the individual is also um, is depleted with dopamine levels, then it could also result to another disorder, which is Parkinson's disease, which is primarily characterized by uncontrollable tremors or uncontrollable movement. No? So you would see shaking and tremors in an individual suffering from Parkinson's disease, and this is something to do with depleted levels of dopamine. Next is serotonin, which has something to do with not just mood regulation, but it's also responsible for eating behavior and body weight regulation. Serotonin is responsible for, as I said earlier, mood regulation, and this is highly associated with psychological disorder, which is major depressive disorder and other depressive disorders, because it has something to do with mood regulation. And so, if you would have depleted levels or low levels of serotonin, then most chances are it would also disrupt your mood, but it will also disrupt uh, eating behavior, sleeping behavior, and also body weight regulation. So this is linked to the deficits or the dysfunction in depressive disorders. Then you also have GABA, which is a major inhibitory um, neurotransmitter. So it has a calming effect when secreted in our nervous system. No? So you have the GABA, the gamma aminobutyric acid. You also have your acetylcholine, which has to do something with learning and memory functions. And so depleted levels or loss of acetylcholine through Alzheimer's disease, no, because of Alzheimer's disease, is linked to impaired memory functioning. So acetylcholine is necessary for learning. Then you also have your glutamate, which is a has something to do with memory, also with learning. And you also have endorphin, which is something to do with euphoria. So endorphin is released when you are involved in physical activity, for example, exercise, yoga. And so it would give you this high or ecstatic feeling. No? So this is what we call the runner's high. No, when you run or when you engage in a physical activity, so you would have this naturally induced feelings of high, no, or elation or euphoria. So these are the different neurotransmitters that we would discuss at least for this lecture. So just a matter of review, no. So this is how it would look like on the synaptic transmission process. No? So first, the electrical signal would trigger when it reaches the axon terminals. No? It would actually trigger the cell wall or cell membrane to open, and it would allow calcium ions to enter, and thus it would also trigger the release of the neurotransmitters, which are packed inside vesicles. So when they are released in the synaptic cleft, they would bind to its specific receptors found in the neighboring or the post synaptic neuron. So when it binds, it creates or triggers another electrical signal, given that the overall or the net electrical signal received from the neurotransmitters will reach the threshold, because again, action potentials, they follow the all or none response. So again, it's because we all know that um, neurotransmitters being received by postsynaptic neurons could either be inhibitory or excitatory neurotransmitters. So it depends on the net or the sum total of the electrical signals, um, whether or not they would reach a threshold that would generate another action potential for the postsynaptic neuron. Now, understanding and gaining information about neurons or about the different regions and structures of the brain um, has come a long way. So there were really an evolution of techniques and methods that they have used before they fully understood about, uh, for example, neurons, glial cells, and the different brain structures and functions. So, for example, for neurotransmitters, for neurons rather, so they would use different staining techniques to see the structure of the neuron, and etc. etc. So for the study of brain structure and its function, again, it has come a long way. So you would really see an evolution of different techniques used in research studies. So they would have to know the structure and functions of the brain. So scientists, they use different methods now for observing human brain and theorizing how it functions. So the the techniques or methods that we would be discussing will be both post-mortem studies, or those involving with dead patients known with different uh, dysfunction or deficits, and later on researchers studied when they died. They studied the brain to reveal what specific structures had damage. They linked it with a dysfunction that they observed when the person was still alive. And we also have in vivo techniques that involves living um, subjects, either non-human or animal subjects, and also living humans. So each technique provides important information about the structure and function of the human brain. So let's first discuss about post-mortem studies again. That has something to do with dissecting the brains of dead patients. No? Now, post-mortem studies usually start during the lifetime of the person. Again, they tend to really select individuals who already manifested deficits or dysfunction when they were still alive. So they don't just study just anyone. No? So it has to be somebody manifesting certain deficits or dysfunction when they were still alive. And after the patient dies, so the researchers would examine the brain for lesions. So, for example, the Wernicke's area and the Broca's area, they were discovered, no, that these parts were actually linked to speech problems, and it has something to do with the post-mortem method used um, by the those who discovered about the Wernicke's area and the Broca's area. So again, the assumption used in this method is that the lesions or damage of a certain structure is associated with the deficit of the individual manifested when he or she was still alive. However, it may reveal the structure, it may reveal the associated parts, for its function because of damage, but post-mortem studies would not reveal um, the function of the brain while it's still alive or while it's still functioning. So again, the post-mortem study has its own weakness, no? Because again, it could not reveal activities of the brain while it's still alive. It could only reveal lesions that can be associated with certain deficits, and so they have to use other methods or other approaches to study a living brain. So another method that they used is on the use or studying of live non-human animals. Now, you may know this that not all methods will be ethically possible for a live human being. So that means that some invasive techniques or procedures will not be ethical to be used in human participants. And so researchers would then be forced or will then have to use um, non-human subjects, for example, primates or cats or rats, in the use of for research purposes. But that doesn't mean that there are no ethical guidelines followed when using non-human subjects. It's just that um, there are more procedures that are ethically allowed, no, to be conducted in an animal than in human beings. No? So again, there are ethical guidelines used even among non-human subjects. So because scientists also want to understand the physiological processes and functions of the living brain, which could not be revealed in post-mortem studies, so they need to use live subjects. So for the study of non-human animals, there are three procedures or three approaches. You have single-cell recording, you have selective lesioning, and genetic knockout procedures. So for the first one, the single-cell recording, it has something to do with the insertion of a thin electrode next to a single neuron. Now, because this is an invasive procedure, this is deemed unethical um, to be used in human participants or human subjects. And through single cell recording, it records the changes in electrical activity that occur when an animal is exposed to a stimulus. For example, an animal may be presented a certain visual stimuli and other kinds of stimuli. So that's for the single cell recording. You also have selective lesioning, which is again another invasive procedure. It is still unethical to human beings. So here, scientists or researchers would purposely remove or damage a certain structure of the brain, and they would have to see what certain functional deficits will they observe once the damage or once the lesion, the removal of that certain part is done. So that's selective lesioning. And you also have genetic knockout procedure, which has something to do with manipulating of the different receptors. So they would have to genetically modify animals so that they will be born without a certain brain cell or a certain receptor, and they would have to see the difference between the function of an animal without these certain receptors compared to those with the normal receptors. No? So these are the different methods used in studying live non-human animals. But then again, one of the major, although it reveals no, the activity, the structure of a living brain, most of the data that they collect from these researchers may not necessarily um, be generalizable to human brains, because again there are still discrepancies with the structures of brain structures of a non-human animal from that of a human being. And so even if it does help us understand fundamental or basic information or basic information about the function of this brain structure, it should still be used with caution, no, when applying to human beings, because again it's not necessarily generalizable, or it's not something the same to that of a human brain. And so we can still use a lot of techniques in human subjects. But again, most are non-invasive techniques, because again, only non-invasive techniques, or those that are really really safe techniques, are only allowed to be used when conducting researches using human subjects. Okay. So, by the way, in studying live humans, there are two major categories or types of neuroimaging techniques. No, so there are neuroimaging techniques that can only reveal the brain structure. That means you can see the different structure of the brain, its regions, but it doesn't necessarily show you the activity levels of the brain at a given time. It doesn't reveal the function of the brain at any given time when presented a certain task. It just really reveals the structure of the brain. And the other type of neuroimaging techniques is to do something with the brain function. So it tells you, it gives you a picture of which area is more active. It gives you the activity level of the brain at a given period at a certain time, and when presented with certain visual stimuli. However, they don't reveal much of the structures of the brain. For example, EEG does not reveal any structure of the brain, while the PET scan or PET scan really has a very, very low resolution of the brain structure. So it could not really reveal a high and in-depth resolution of the brain structure. And so both types of imaging techniques are used by researchers and even physicians and and other professionals in studying a patient's brain. Okay, but there are also more recent or advanced studies that can combine both, or can both yield the brain structure and the brain function of an individual. And one technique is the functional MRI, or the functional magnetic resonance imaging, which can actually reveal the brain structure and also the brain function, or it could also reveal which parts of the brain are most active in this specific activity. Okay, so again, they are used depending on the purpose. So let's first discuss about a method of studying live humans rather, that has something to do with recording the electrical activity of the brain. And so the brain, when it processes things, even if you're not necessarily processing something, it would simultaneously and would continuously send electrical signals. And so the one technique used, is to record electrical activity of the brain. And one example that you can use is the electroencephalogram, or EEG, which gives you the overall electrical activity within the brain. And this is essential because it reveals to you different states. For example, it reveals to you when an individual is um, aroused, or when an individual is sleeping. It can also reveal to you when an individual is experiencing a seizure, or it could also reveal different stages of dreaming. No? So again, it can precisely tell you the different electrical changes or activity level at any given specific time. However, a weakness for this one is it does not tell you about the structure of the brain. It will just give you the graph, about the electrical activity. Next is static imaging techniques. So aside from electrical levels of the brain, other methods could also use to reveal pictures or still photos of the brain. So you have your CT scans, you have your MRI, used for seeing the structures of the brain. But again, it just gives you the structure of the brain, but it cannot reveal to you the current activity level of the brain, which areas of the brain are most active, which are not, which are less active in this specific task, and etc., etc. But it can actually aid physicians um, to diagnose certain brain disorders. For example, whether the person has tumor, or are there abnormal growths in the brain, and things like that. Because again, it reveals the structures of the brain. Um, so here are some examples of the um, different techniques used to study the structure and functions of the brain. So you have your brain angiogram, you have your CT scan, you also have MRI scan, PET scan, and TMS. And you also have another method, which has something to do with metabolic imaging. So this one tends to be, or can be, invasive, but it is relatively really safe. For example, for the two examples of metabolic imaging are PET scan, or positron emission tomography scan, and fMRI. For PET scan, radioactive substance is either swallowed by the participant or either injected. It's called a tracer substance, and so it can bind to different molecules in it's injected. No, it's ingested in your bloodstream, and it binds to certain molecules, and it reveals. It can give you a picture of which parts of the brain are most active in a certain activity. Okay, so it has something to do with the consumption of glucose and oxygen in active areas, because areas in the brain which are primarily involved in that process, they need more energy, so therefore they need more glucose and more oxygen, as compared to those areas which are not directly involved in that process. For example, when you are looking at a visual stimuli, you would expect that your primary visual cortex or your occipital lobe will be the most active. So therefore, if you look at it in a PET scan or fMRI, you would see there that it's your um, occipital lobe, which is highly active in that participant activity, and it has something to do with the increase of consumption in glucose and oxygen. Yes, so that's for metabolic imaging. So it's only the PET scan that uses the a radioactive substance, so that it could reveal the different active areas. But for fMRI, it wouldn't require any tracer substance or radioactive substance to reveal the active areas in the brain. So it can reveal both the structure of the brain. It also reveals which areas of the brain are most active. However, it tends to be slower. fMRI tends to be slower as compared to EEG. So there could be like very minuscule changes in the brain activity, which could not be detected by fMRI, but could be detected by EEG. So again, they are used together. No? So when studying a human brain, so they can be used together. So here's a table. This can be found in your book about the different methods or the procedures of studying the human structure and the function of the human brain, and it also discusses about its advantages and disadvantages. So I want you to read each methods, its advantages and disadvantages. For example, for single cell recording, for EEG, for ERP, for PET scan, for fMRI, for TMS, MEG, um, FTCD, and near infrared spectroscopy. So they have their own strengths and they have their own weaknesses. For example, single cell recording could only be used for non-human subjects because they are invasive, while the rest can be used with human participants. Some can be cheaper, some could be more expensive. For example, EEG is cheaper compared to MEG because MEG, although it gives you a precise resolution, no? But it requires a very expensive machine and it requires also a specialized room so that you can perform or you can use this machine or technique. But EEG is relatively cheaper, no? And it still has its own value. Okay. Now, Now, neuroimaging techniques are not only essential, no, in uncovering different structures and functions of the brain that would further the understanding of scientists, of researchers. So it's not only for research purposes. Neuroimaging techniques are also very essential to aid diagnosis for certain brain disorders. And we all know anything that damages the brain would also lead to certain deficits and dysfunction within an individual. And so neuroimaging techniques help medical doctors to see whether a stroke has occurred or whether a tumor is present in an individual's brain. Because again, brain disorders can impair cognitive functioning, and it gives us valuable insight into the functioning of the brain. So we would only be discussing three brain disorders or three um, things that would also impair cognitive ability, but there are a lot other there are other brain dysfunction that you can also see, but it's not here in your book, so we'll only have to cover three. No? So first is stroke. So stroke is a vascular disease or a vascular disorder um, that is a result of the disruption of blood. No? So when there is a obstruction, so if there's blood could no longer flow, then it could lead to stroke. So people who experience stroke may show marked loss of cognitive functioning. So speech is slurred, Even motor movement is also affected, and other cognitive functioning. No? Memory, there are cases wherein some mini strokes would mimic the memory loss in Alzheimer's disease. And so again, it really would impair an individual depending on the amount of damage it um, affects the brain, or the amount of disrupted area in the brain. So stroke could either be ischemic or hemorrhagic stroke. So there are different two types of strokes. No? So a person experiencing stroke may be paralyzed, may be in pain, may experience numbness, loss of speech. Again, has to do something with language comprehensions and speech, and a lot other um, impairments in cognitive function. So, how can an individual, or what could be the symptoms of an individual having stroke? No? So, there is number one, numbness or weakness in the face, the arms or legs, and it's usually only just in the one side of the body. No, so because stroke usually occurs in one part of the brain, then the opposite side of the body will be affected. No, so if you can still remember the ipsilateral and contralateral processing, so there are times wherein information is processed within the same side of the body. For example, information coming from the left side of the body will be processed by the also the left side of the body. While there are also times wherein contralateral processing occurs, wherein the right side of the body, information coming from the right side of the body will be processed on the left side of the brain. So that's the contralateral processing. There's also confusion and difficulty in speaking or understanding speech, vision disturbances, dizziness, trouble walking, or loss of balance or coordination, and severe headache with no known cause. And it's very important class to for an individual experiencing stroke to have immediate or to receive immediate medical attention, no? Because it can lead to brain damage, no? Permanent brain damage, long-term disability, and even death. So it's something that needs immediate medical attention. You also have brain tumors, are also called neoplasms, and it is also very disruptive to your brain again because it can damage as the tumor grows bigger. No? It can damage surrounding tissues or surrounding neurons. No? So therefore, if it damages an area, it could also damage the function um, involved in that area. No? So for example, if you have a tumor responsible for your motor movement, so motor movement will also be affected. Um, so brain tumors can be either cancerous or benign. They can be life-threatening. So common symptoms for brain tumors could be headaches that is usually worse in the morning, nausea and vomiting, changes in speech, vision, or hearing, problems in balancing or walking, changes in mood, personality, or ability to concentrate, problems with memory, muscle jerking or twitching. There could be seizures or convulsions, and numbness or tingling in the arms or legs. It doesn't mean that if you have these symptoms, you already have brain tumors. So, but if you do, and if it hinders you to do tasks, then it's better to seek professional help. And lastly, you have head injuries. No, so now if you can still recall the topics in physiopsych, about the different defense mechanisms of the brain, you have different structures that would protect our brain from threats. No, so you have your external, you also have internal defense mechanisms of the brain. No? So for external, you have your skull, a hardy case bone enclosure that would protect you from outside threat. For example, if somebody knocks you out, or if you hit your head, so it would protect your brain. And aside from the skull, you also have your meninges, which are underlying structure under your skull, and even your cerebrospinal fluid that acts as cushion. No? If ever you hit your head, no? So it's one line of defense in your brain. You also have your blood-brain barrier, which protects you from internal threat, for example, from harmful substances. So it does not allow the passage of harmful or non-essential substances. It's only glucose, oxygen, and other um, substances necessary for brain function are allowed to enter. But psychoactive substances could easily penetrate the blood brain barrier. And so, head injuries could be a result from different accidents. No, it could be from a car accident, it could be from hitting your head too hard, maybe you slipped and you hit your head at the floor, and things like that. So, head injuries could be closed head injuries or open head injuries. Closed head injuries happens or happen when the skull remains intact, but there is brain damage. And open head injuries is when the skull is penetrated. No? And of course, there is brain damage. So, a loss of consciousness is usually a sign that there has been some degree of damage of the brain as a result of the injury. So, not every time that you would hit your head, it would necessarily create damage to your brain. No? Depending on the impact and on the intensity, no, of the concussion. For example, if you hit your head in the jeep, diba? If it's not too hard, then you're fine. But again, if you if you would lose your consciousness, then it may tell you that maybe there is some damage to the brain. So damage resulting from head injury can include spastic movements, difficulty in swallowing, slurring of speech, um, other cognitive problems, and even death if it's too much. No, so those are the different um, consequences to head injuries. And so basically, this lecture tells us, or to summarize this lecture, it tells us about the different regions of the brain, how these brains interact with each other, how different brain regions also have their own specific function. They have their own specific, they process specific information. Um, Neurons also play a part in communicating with each other, in transmitting, in sending and receiving signals. Your neurotransmitters are essential to um, passing the signal from one neuron to another. We also learned about the different methods of studying the human brain, from post-mortem studies to studying um, non-human subjects and also studying live humans. You also have different techniques used in studying human subjects. You have your electrical recordings, you have your static imaging techniques, you also have your metabolic techniques used in understanding live human brains. And we also tackled about different brain disorders and how it can also affect cognitive functioning. And so hopefully you were able to understand most of the concepts for this chapter, which is cognitive neuroscience. If you have any questions, it's better for you to have your own personal reading and also to consult other sources in reinforcing your learning. Thank you guys for listening, and I hope you're safe.